This presentation will show you how to extract DNA from a strawberry. In other words, how to take a strawberry that you would normally find at the grocery store and be able to access all the DNA within its cells. To start off, we need to have specific materials to help with this DNA extraction. Looking at this image and starting from the left and working to the right, we have here a funnel which contains gauze, but cheesecloth can also be used, placed in a round bottom flask. Next to it, I have a tall graduated cylinder containing about 90 milliliters of distilled water. I have meat tenderizer, and then I have a graduated cylinder which contains about 20 milliliters of isopropyl alcohol, which is the same as rubbing alcohol. Then I have a smaller graduated cylinder containing about 10 milliliters of dish detergent. In front of all of this glassware, I have 2 strawberries and a single test tube. The first step of a DNA extraction is to take the specimen that I'm using containing the DNA, so that would be the strawberries, and placing them into a Ziploc bag. I add a pinch of meat tenderizer, the 10 milliliters of the dish liquid, followed by the 90 milliliters of the distilled water. What these items do is create a DNA extraction solution. The dish liquid is meant to be used to break down the cell membrane, thus opening up the cells and also breaking the nuclear membrane, which also opens up the nucleus in order to release the DNA. The meat tenderizer has special enzymes or proteins that can break down other proteins that keep the DNA in place. And then the water is what puts everything into a solution. So when the strawberries are submerged in the extraction solution cannot properly access every single cell that makes up each strawberry. The strawberries have to be smashed open so that way the extraction solution can accurately open up and break all cells within the strawberry material. After smashing the strawberries using the extraction solution, it must sit for about 5 minutes. This allows the materials within that solution, also known as the dish liquid and the meat tenderizer, to actually be able to break open the cells, break open the nucleus, and break down the proteins that hold the DNA in place, ultimately releasing the DNA into the water solution itself. The problem is that there are chunks of strawberry mixed in with the DNA dissolved within the water solution. This is where the gauze in the funnel is able to separate the solid pieces from the liquid solution. So by slowly pouring the solution into the funnel containing the gauze, we'll catch the solid pieces and allow all of the liquid to pour through the funnel into the round bottom flask. It is within the liquid that the DNA will be found. As this process happens, the liquid containing the DNA will fill up the bottom of the round bottom flask. Ultimately, the round bottom flask will contain the solution with all of the extraction items as well as the DNA. To better manipulate this solution and to more accurately separate the DNA from the rest of the solution, some of the strawberry with extraction solution was poured into a glass test tube. In order to separate the DNA from its solution, isopropyl alcohol, which has been cooled, will be added to this solution. What it will do is actually separate the DNA and allow the DNA to float to the very top. On the next slide, there will be a video that will show lots and lots of DNA forming a globule floating to the very top of all of the liquid. Pay very close attention as you watch this short video so that way you can see this somewhat clear whitish material clumped together and float to the top. That will be the DNA. At the end of the DNA extraction, looking into the test tube, you can see that at the top of the solutions would be a chunk or a glob of white, somewhat clear material. This material is in fact the DNA. Remember, we broke open many cells that make up each strawberry, thus releasing many pieces of DNA, which normally would not be able to be seen without the use of a microscope. All these pieces of DNA came together to form this giant glob. This is why at the end of this extraction, we are able to see all of that DNA, because those are DNA molecules, which are thousands of molecules from thousands of cells.